Well, good morning, everybody, and it's a real pleasure to have the opportunity to talk to this group. I really would like to thank Mark Miller and Melanie and others from OEHA to make this invitation to me. I, as you've already heard, haven't really been so involved with color additives over the last 6 or 7 years. So it's been kind of interesting to get back into some of the data and take a look at sort of what's already been reviewed by the FDA, but also to look a little bit at what some of the new information is that's out there now. And so what I hope to do this morning is just give a quick, relatively quick outline of how some of these color additives were first evaluated by the FDA. Ask, I think, a relevant question, which is, are these evaluations out of date at this point? And then also maybe look forward a little bit as to where this might be going. So colors, you've already heard from several people, have a long history of safe use here in the United States. Colors provide aesthetic value. Obviously, I think most of us have probably eaten a red velvet cupcake or something with a lot of blue frosting or orange frosting or yellow frosting at some point. But they also do other things. Colors are really important in the pharmaceutical market to help with appropriate identification of pharmaceuticals, pills in particular. This is an especially important problem for the aging population, where mistaking of certain pills has led to absolute serious health consequences. So making sure that the red pill is appropriately red, the blue pill is appropriately blue, is really important, and color additives play a critical role there, obviously. Consumers have an expectation for how their foods are going to look, and so a raspberry flavored food typically is expected to have red color. A blueberry flavored food, maybe a blue or purple color. And without that, there are a lot of studies that show that consumers, in fact, are often very comfortable rejecting those foods outright. And then finally, colors play a pretty critical role for the food supply in that they offset the color loss that often occurs just from either natural color variation, so the natural inherent color, say, of orange juice or of other types of foods is lost because of light, heat, oxidation, long-term storage. And so in many cases, colors provide that role to help supplement that color and ensure, again, that consumers have the opportunity to have the color they expect. Dr. Lippman has already talked in depth, I think, about the differences. Certified colors, just to remind you, many of these were originally isolated from coal tar oil sources. Now, mainly these are produced from relatively simple chemical building blocks. Each batch is tested by the FDA to confirm safety and use. So there's an FDA color certification lab, and every color manufacturer has to submit a sample of each batch into the FDA. It's a fee for service, so the industry pays for this to be done to ensure that the color has appropriate specifications. The impurities are controlled, et cetera, below a certain level. These are, again, the FD&C color, food, drug, and cosmetic colors. The exempt from certification colors on the other side, many of these, I should say, were originally isolated from botanical sources. These are things like grape skin extract, grape seed extract. These do not require batch testing. These are all color additives, though, as we'll discuss, and so they all require some sort of safety considerations. Dr. Lippman has already outlined colors are food additives. This should say 1938 Food, Drug, and Cosmetics Act. 1958 was the Food Additives Amendment to the Food, Drug, and Cosmetic Act. 1960 was the Color Additives Amendment. All color additives, as a result of that 1960 Color Additives Amendment, require pre-market approval via the Color Additive Petition process, which again involves a safety assessment and review by the US FDA. And those colors that have made it through that process are listed under the U.S. Code of Federal Regulations at Part 21, Section 73 and 74. I guess I would say the one potential exception to this are the fruit and vegetable juice colors, where for those that are from the ripe, mature vegetable or fruit with very minimal processing that would use only water, these don't require a traditional color additive petition process. In the US, all color additives are considered artificial for labeling purposes. In other words, if you're using Blue 1 or Blue 2, or whether you're using blueberry extract, if you're putting it into a food, it's still considered an artificial coloring. And so it requires some sort of labeling. For the FD&C colors, that means those colors have to be listed by name on the ingredient statement. As Dr. Lippman's outlined, carmine cochineal also, which is not one of the FD&C colors also is required labeling due to the allergenic potential. And I think what this means is there's not a lot of ambiguity here in the US. So colors, especially the certified colors, are labeled. And if a consumer is not interested in consuming those, all they have to do is flip that jar around and take a look and decide whether, in their case, they would like to buy this product or not buy this product. So how do we get there? Well, so the FDA Red Book, which I think Dr. Voorhees has provided a nice preliminary look at this morning, recommends toxicity testing based on risk concern levels. And so this list up here gives you a rough idea. Concern level low, in other words, substances for which the FDA has relatively minimal ingoing concerns about the potential toxicity or adverse health effects, typically requires genetic toxicity testing. These are tox tests that probe the ability of a substance to cause some sort of DNA damage, maybe DNA mutation, possibly chromosomal damage, as well as, for concern level low substances, short-term toxicity tests with rodents. Concern level intermediates, these are the ones that, for which maybe the FDA has said, well, we aren't going to go in and say that these things are likely to be safe or likely to have low potential for adverse health effects. We're going to ask for a lot more information here. And in this case, they're asking for subchronic toxicity tests. These are usually 28-day tox tests and then 90-day toxicity tests with rodents, so rats, mice, with non-rodents, rabbits, beagle dogs in many cases. And then other things, reproductive and developmental toxicity testing, metabolism, pharmacokinetic studies in some cases, and any other special studies that the FDA would think would be appropriate. A lot of this testing process is done not directly in collaboration with the FDA, but under some form of FDA guidance. And so it's very common, for instance, for somebody who's interested in a new food additive or color additive to go to the FDA at an early stage and get feedback as to what sort of testing might be helpful and required for the FDA review. And then those concern level high ingredients are the ones for which additional elements are required. And those, in many cases, are 1-year toxicity tests, and then what we refer to as either chronic tox or carcinogenicity testing, or a combination of both. And those are usually in 2 rodent species, rats and mice. They're typically 2-year administration studies, repeat dose, where the animals receive the dose either every day or typically 5 days per week over a 2-year period. And then in all cases with these studies, there's extensive analysis to understand what potential effects are observed. Now, this testing, essentially all of this testing that you're looking at here, perhaps with the one exception of the human studies that are looked at up here, are usually done under some sort of standard guideline. So internationally, there's the Organization for Economic Cooperation and Development that Dr. Voorhees mentioned this morning. The OECD has a whole list of standard guidelines to do toxicity testing, chemical safety testing. They largely align, I would say, with the FDA's approaches here. In Japan, similarly, there's a set of guidelines. Sometimes those Japanese guidelines vary very slightly. Could mean a different strain of rodents would be preferred versus others. In Europe, they usually are interested in applying the OECD guidelines. So how do we go from recommended toxicity testing to the actual data that we have, the tox data that we have for colors? Well, there's a substantial safety data set available for many of the colors. The FDA review led to listing within the Code of Federal Regulations based on all of the data that I'm going to be showing in just a few minutes, with a few exceptions. So there's new data in some cases that hasn't been reviewed by the FDA. Over the years, once these color additives have been listed by the US FDA, there's additional genotoxicity, in other words, assessment of potential DNA damage. There's in many cases allergenicity studies that have been done, some toxicity studies, and in some cases reproductive or developmental tox studies have been done. In many cases, but not all, these are based on requests from regulatory bodies. So in some cases, the European Food Safety Authority, for instance, would say, thank you for submitting this package of data for FD&C Red number 40, which when not certified is referred to as Allura Red. But we would also now like to see an assay of DNA damage in rodents, in mice or rats. So please do that study, submit it to us. We'll let you know if we have additional concerns after we review it by publishing an updated opinion. In other cases, when the industry has identified the need to do additional testing, again, it really has primarily tried to rely on these standard guideline studies. One of the challenges, I think, for industry to do a new study is not necessarily the will or the interest, but it's the question as to how widely accepted that study would be. So if you stray too far away from a standard guideline, there's always the question as to whether this would be accepted in Europe or by the US FDA or others. And this remains a challenge, I would say, because there is harmonization for a lot of the standard tests, but as we already heard this morning, there's a lot of interest in developing new test types. I think that's very interesting and exciting. It would be very attractive if we could get to a place where there was a relatively standard and agreed upon approach to do that type of testing. So jumping into the tox data, as I was asked to take a look at. So what you can see here, these are the azo dyes, the FD&C red number 40, FD&C yellow number 5, and FD&C yellow number 6. And there is a lot of information up here that I don't intend to go through line by line by line by line, since it would take quite a long time. But what I wanted to try and do here is give you a rough idea for what the FDA has generally looked at when it evaluated these within the context of the color additive petition process, and also point out some places where there's additional data that's now become available that the FDA hasn't necessarily seen or considered within the direct context of the color additive petition process. So red 40, as a first example, really has been in use provisionally since about 1971. There is negative in vitro and in vivo genotoxicity data. What that practically means is, back when these color additives were first listed, there weren't a lot of great tests for gene tox assays that were out there, or I should say for DNA damage that were out there. Over time, however, there have been the development of OECD standards for gene tox testing. There's a DNA mutation test referred to as a bacterial reverse mutation assay, or an AIMS assay. That is now one of the preliminary initial screening assays for mutation. Allura Red AC is negative in that assay. There's also several different chromosomal damage studies in vitro and in vivo that are now available, for which there are standard OECD guidelines. And these include the in vitro micronucleus assay, the chromosomal aberration test, and others. Allura Red generally has tested across the board negative in those assays, with one exception, and that is in the COMET assay. COMET assay is a relatively recent test for DNA damage. It's a screening assay. There is an in vitro way to do this test, but the in vivo study is much more commonly performed. And over the years, there has now been the development of an OECD guideline for a COMET assay. So I think back in about 2002, there was a paper published by Sasaki et al. From a Japanese lab, where they collected a positive outcome in a DNA damage assay, a COMET assay for Allura Red AC. That was or I should say prior to the development of the OECD guideline, prior to the validation efforts to show that any lab could do the COMET assay in a way that would allow for reproducible and reliable results. Since the publication of the OECD guideline assay, the industry, at the request of the European Food Safety Authority, conducted a new COMET assay for Allura Red AC and collected negative results. As a result of that, EFSA concluded, in their eyes, that the earlier results from Sasaki et al. Were probably not relevant. And therefore, the OECD standard guideline study was accepted by EFSA, and it was concluded that this substance doesn't possess genotoxic activity. You can see the rat, dog, and pig acute subchronic studies have been done with quite high NOEL values. The chronic studies that have been done for rats and mice have similarly shown significantly high no observed adverse effect levels, up to 7,300 mg per kg body weight in mice and 901 milligrams per kilogram body weight in rats. Those of you in the tox community, these numbers probably look really pretty high compared to especially some of the pesticides that maybe you see on a regular basis. This is not that uncommon for food additives. I would say in general, food additives, by the time they get to the point of a color additive petition process or a food additive petition or a submission to the European Food Safety Authority or JECFA or others, they're generally not that toxic. The ones that show significant toxicity are usually the ones that don't make it through this process and even get to the point for regulatory review. We've talked a little bit this morning about some of the neurobehavioral studies. These have not been evaluated by the FDA within the context of the color additive petition process. There were, I think, as a result of the literature search related to the 2011 FDA Food Advisory Committee meeting, interest in evaluating the Tanaka et al. Study that Dr. Gall, you described this morning. Since that time, there have been additional studies. And I think Dr. Gall, you talked a little bit about some of those. The Doguch-Chahan study, I think from 2013 and 2015, that also probed some of the neurobehavioral effects. And again, the Southampton study from Stevenson et al. Looked at hyperactivity effects. That was looked at, of course, within the context of the 2008 Citizens Petition to the US FDA and the 2011 Food Advisory Committee meeting. To the best of my knowledge, the US FDA has not reviewed the Loke et al. 2013 study, which was an attempt to reproduce the effects from the McCann et al. Study from 2007. But certainly I think that would be of interest to them potentially. And we talked a little bit this morning about ADME, absorption, distribution, metabolism, excretion, and potentially toxicokinetic or pharmacokinetic data. And I fully agree that there's a lot of interesting questions here that haven't been fully explored. And it would be interesting, I think, to better understand some of the more detailed toxicokinetic profiles for some of these ingredients. What I will say is that at the time when some of these initial provisional color additive listings were made and color additive petitions were filed, the understanding of how to do such studies was pretty limited. And so I think it's an interesting time. I think we could probably do more here. And I think it would be interesting to take some of that on. What we can say is, for Allura Red AC or FD&C Red number 40, in general, as I think Dr. Marty mentioned this morning, the absorption is pretty low. So in this case, 5 to 8%. We do know a little bit about the metabolism. We know that these things do undergo intestinal reduction, so azo reduction. Whether the potential aromatic amines are, the extent to which they're absorbed, the extent to which they could perform other potentially adverse effects is an interesting question. Aromatic amines, I think, in the eyes of the tox community, raise obvious questions and concerns about possible carcinogenicity. And what we've seen from 2-year CARC studies, as is shown in this slide, is that we don't see those effects here. So these substances don't seem to be carcinogenic. They don't seem to be genotoxic. Even if the metabolism is leading to the formation of small amounts or significant amounts of the aromatic amines, we're not seeing obvious indications that this is a particular toxicological concern leading to an adverse health effect. And then at the bottom of the slide, I've thrown up the JECFA ADI value for FD&C Red number 40, and that's 7 milligrams per kilogram body weight. So that's one example of the type of data that was looked at in the original Colorado petition, along with some additional studies that have either now been looked at by the FDA within the context of the 2011 FDA Food Advisory Committee meeting and the attendant report, or in some cases data that I don't think the FDA has yet seen. We could do the same thing for tartrazine, for sunset yellow. I think what I want to draw your attention to, though, is that in general, the profile that we see for the FD&C colors is that they don't exert significant DNA damage effects. They're negative in vitro and in vivo gene tox studies. The available data in subchronic or chronic studies generally indicates a very low order of toxic potential. So in a repeat dose, repeat administration type study, typically done via dietary administration, in some cases via encapsulation, so they put the dye inside a capsule, in some cases just by directly adding it into the feed. They're not exhibiting significant amounts of adverse health effects. Those are the azo dyes. Moving on to the blue 1 and green 3. When these are not certified, these are referred to as brilliant blue FCF and fast green FCF. Again, these have been in use for a very long time. They were some of the early ones that were provisionally listed and then the subject of color additive petitions. And you can see here that these also are generally negative in genotoxicity studies. In chronic or subchronic toxicity studies, they show a very low order of toxic potential, no evidence of carcinogenicity whatsoever. There are repro and developmental tox studies for which NOELs have been established. And then additional studies, in some cases the neurobehavioral studies have been done, the Tanaka et al. And others, And then available ADME data, once again, suggest a very low order of absorption, so generally not particularly bioavailable. And we have some understanding of the metabolism of these. In these particular cases, I think we see, for these particular dyes, a little bit less likely metabolism and a lot more excretion of the dye directly. And then finally, the last two, indicating and erythrocene, when certified, FD&C blue number 2 and FD&C red number 3, again in use for a very long time. Again, very low or no expectation of genotoxic potential. In other words, no likelihood of DNA damage. Acute, subchronic, and chronic toxicity studies indicate generally a fairly low order of toxicity. I will draw your attention to the fact, however, that for erythrocene, this is probably the dye with the most toxic potential. It's not substantial, but it's still there relative to the others. The others are very, very low order of toxic potential. Erythrocene is a little bit different than that. Additionally, erythrocene has been previously flagged in carcinogenicity studies. It's shown under dermal applications to cause thyroid cancers. That's not an effect that is observed upon oral administration. So it's really only a dermal effect. And because of that, the FDA limits the way that it can be used. Again, reproductive teratogenic studies indicate valid NOELs using standard guideline studies. It's clear indication from the ADME available, the data that's available in ADME studies, that these are, again, also very poorly absorbed. So I don't really need to spend a lot of time here. I think Dr. Voorhees has already gone through, and Dr. Lippman as well, the additional considerations that the FDA has looked at with these color additives, including, again, the results from the 2011 FDA Food Advisory Committee meeting. They've also, within their report and their background reading information, provided acceptable daily intakes within that report, which I'll mention in just a minute. The International Association of Color Manufacturers has continued to collect relevant data. This is toxicity data, reproductive tox data, genotoxicity, and more recently has published a paper related to exposure assessment, which I think will be discussed perhaps a little bit within the context this afternoon of what the FDA has been doing. So one of the questions here is, are the FDA's conclusions out of date? So we've just reviewed these. I've already said, they've looked at a lot of these a long time ago. They looked at the hyperactivity issue maybe a little bit more recently. But you might say, well, if it's been so long since these were provisionally listed, has the FDA looked at them more recently from a safety perspective? And I think the simple answer to that is no. They haven't picked them up recently. There hasn't been an effort to do that. There hasn't been a petition filed to suggest that there are specific safety issues related to these color additives, other than, of course, the 2008 citizens petition that was filed. But you could look at it from a different perspective and say, well, what do we know about what others have been saying about these color additives? Are there more recent safety assessments for these colors? And of course, this has already been discussed a little bit, but the UNFAO-WHA Joint Expert Committee on Food Additives, the JECFA, has more recently reevaluated FD&C colors and confirmed the safety and use. So in 2011, they looked at FD&C yellow number 6. In 2016, at red 40 and yellow 5. In 2017, blue 1 and green 3. And in 2018, they looked at blue 2 and red 3. And you can see in all cases, JECFA concluded that the ADI should be either retained at the current levels or increased or decreased. So there were some changes here, but nothing really substantial that I would say. And of course, JECFA has already been mentioned, has also considered the McCann 2000 data on the azo dyes and the mixtures of food color additives, and come to the conclusions, which I'll just hit the highlight here, that these studies are of limited value within the context of the JECFA safety assessment because of inconsistencies in the findings and the use of mixtures of food colors. It doesn't mean that these aren't important and interesting studies that are worth looking at, but within the context of what JECFA has done here, they've concluded that they're not going to use this within the context of their safety assessment. EFSA has also reevaluated these colors and also confirmed the safety and use. And again, these are very recent evaluations. So 2014, 15, 16, 12, 11, et cetera. So very recent publications, which again provide conclusions that these are safe in use and also provide updated acceptable daily intakes. And of course, we've already talked a little bit this morning about EFSA's conclusions, where the EFSA food additive panel, assisted by experts in behavior, child psychiatry, allergy, and statistics, provided their conclusions about the use of the McCann data. And I'll jump through there to my next point, which is, I think what this is saying is if you look at the ADIs developed by the FDA, by EFSA, by JECFA, they're all generally in the same order of magnitude, with the one difference, of course, being erythrocene, where there are some differences in the ADIs that have been established for erythrocene. And that's, of course, based on different interpretations of no observed adverse effect levels. And they've all reached generally consistent conclusions about the available data relevant to hyperactivity. So I know I just have a few minutes left. I want to talk a little bit about what the future might look like here. So the FDA Red Book, as Dr. Voorhees described this morning, provides some preliminary guidance for the consideration of neurotoxicity. And I have to say, I'm absolutely not an expert here, so would absolutely yield to those in the room that are. But my take on what the FDA Red Book is saying here is that you start with screening to identify chemicals that exhibit potential for adverse nervous system effects. One way to do that is through the use of structure-activity relationships. My limited understanding is that there really aren't good structure-activity relationships for neurotoxins at this point. Second way is to say, well, what's out in the literature? Are there evidence or are there published reports that suggest that there are clear neurotoxic effects here? And the third would be empirical testing. And from that screening, if you see neurotoxic potential, then you do more work. From that screening, if you don't see it, then maybe that's not a sufficient basis to do anything further. I think this is my takeaway from what the FDA is saying. I think what we heard this morning from Dr. Voorhees is maybe that paradigm needs to be considered in the opposite direction. So you start with the observation and you work back to consider what would be available from the screening type information. And if I'm wrong, please feel free to correct me on that. So empirical data, well, you can do animal studies. We've talked already this morning about the limitations of FOBs. What I can also say is that if you look back at the old subchronic and chronic tox studies for the color additives, it's crude observational information at best. And from the histopathology and pathology work that was been done, they did brain tissue, they looked at the brain tissue, but that's a lot different, I think, than what we're talking about in terms of state-of-the-art study these days. The other alternative is high-throughput screening. And here, we do begin to have some data from tox testing in the 21st century. So ToxCast, Tox21 program from the US Environmental Protection Agency, US FDA, the National Institutes of Health, and others have begun to do high-throughput screening assays against a very large number of chemicals, and fortunately and interestingly, for some of the food color additives that we're talking about this morning. And so this is work now that I'll show very briefly that's still under development. This is work that's been done by Tox Strategies, which is a scientific consulting firm. I've had a conversation with them and was able to get to the understanding, I think, of what they're trying to do here and where this might be going. So they're interested in a tiered approach. So let's see if any of these food colors are on known neuro lists. In other words, neurotoxins that are already identified. Let's see what the ToxCast data holds. Let's look at animal studies, the relevance of those studies, and let's look at human studies. Tier 3 and Tier 4, I don't think we're going to talk about this morning. That's already been described in detail. But Tier 1, this very crude screening assay, there doesn't seem to be any indication that these are expected to be neurotoxins based on the available databases of expected neurotoxins that are present within the EPA computational toxicology database. What does this mean? From my perspective, it doesn't mean very much at all. So these are not going to kill you right away. These are not cocaine. These are not methamphetamines. There's nothing surprising here, right? It's just an opportunity to say, well, they're not hitting in any of the traditional computational toxicology databases. On the other hand, what is a little bit interesting, I think, is that if you go into that ToxCast Tox21 database and you look at assays with neuro-related adverse outcomes, what we see is that of the 112 assays for which there are, let's say, neuro potential adverse effects that have been done specifically for these 7 food colors, 7 of them show activity. In other words, the very vast majority of the assays done with these food colors do not show any sort of adverse effects or hits, let's say, in ToxCast Tox21. This is, of course, preliminary analysis, but I think one of the other things that's pretty interesting here is that the one that does is erythrocene. So of the 15 assays that have been looked at, erythrocene shows hits on 4 of the 15. Allura Red, which is one of the azo dyes, doesn't show any. Tartrazine shows 1 out of 21 assays. Sunset Yellow, 0 out of 23. So all of this data that's now being produced by the US EPA, by the FDA, by NIH, by others, is giving now, I think, a first screening picture as to where we might want to look further, where there could be some interesting questions to address. And I think, given my time limitations here, I think one of the things I'll say is there's a lot of work to be done here. But when we look at what hits we have for erythrocene, these are relevant receptor or gene targets, but they don't show causation. What they show, I think, are potential opportunities to explore further. So these gene measures that were being looked at here, they're informative for studying mechanisms, but they do not independently indicate hazard or risk at this point. So I think they raise some interesting questions for exploration. I think additionally, the fact that the traditional azo dyes and some of those that were used within the context of the Stevenson et al. Study do not seem to have a direct molecular correlate, at least within the ToxCast Tox21 database, I think is really interesting and worth exploring further as well. So with that, I'll stop now.